Welcome. I'm Loni Schumacher, a veterinarian at the K-State Swine Nutrition Team, and thank you for listening to my second Midwest Meeting podcast entitled Evaluating the Effect of Manufacturing PED Contaminated Feed on Subsequent Feed Mill Environmental Surface Contamination. This presentation is a complementary study to my previous sequencing presentation, where in this study we evaluated the feed mill surface contamination following production and sequencing of a PED-infected batch of feed. If you can recall in our previous research, we established a minimum infectious dose. Note that the infectivity of the virus in feed is very low. The current study is an observational study that complements my previous podcast called Utilizing Feed Sequencing, where we are looking at feed mill surfaces and where potential contamination can occur on these surfaces and whether sequencing will eliminate the contamination. Thus, the objective for this study is to evaluate the potential for environmental PED contamination in a feed mill after PED inoculated feed was produced. Again, this was done at the FSRC, which is connected to our feed mill and is a pilot and contains pilot scale equipment. Our methods for this is the same as the previously presented in the Utilizing Feed Sequencing podcast, where we made negative, positive, and 4 batches of sequenced feed. The only difference for this complementary study is that we collected swabs from designated locations after each feed treatment was mixed and discharged. In this study, we were looking at the environmental and surface contamination that ensued. For our materials and methods, we used swabs that were pre-filled with 2 mils of sterile PBS. Baseline samples were taken before each repetition, and swabbed areas were outlined, 5 subsampled areas that were equal in size but ranged between locations. Subsampled areas per location were randomly allocated and sampled after each feed treatment. Thus, no exact spot within the subsampled area was sampled twice. Zoning is a systematic method of pathogen sampling, and in our study we looked at 3 zones, Zone 1, 2, and 3. Zone 1 is the direct feed contact surfaces. Zone 2 is immediately adjacent to direct surfaces. And Zone 3 is outside Zone 1 and 2, but still within the facility. Swabs were taken after each feed treatment, and here is a list of the zones and the swab areas that we took per zone. In Zone 1, the mixer, bucket elevator, and pellet mill. In Zone 2, high and low foot traffic, drain, metal table workbench, and also the bottom of the rubber boots. And finally, in Zone 3, we sampled the garage door. Here's how we categorize the swabs according to zone. Zone 1 consists of the pellet mill, bucket elevator, and mixer outlined in red. Zone 2 is the table or workbench, high foot traffic area on the floor, the drain, and the low foot traffic area on the floor, highlighted in orange. Zone 3 is highlighted in green and is the garage door that's directly behind me as I'm taking the picture. Here are the results, the effects of PED sequencing on the zone CT values. Zone 1, as you can see after the positive feed was made, and as surfaces were positive over time, the surfaces decreased slightly. This makes sense because during sequencing, we have clean feed that's diluting out the detectable RNA. In Zone 2, in orange, all surfaces were positive and remained so throughout the study. Again, we did not clean surfaces between treatments, so that if RNA was present, it would remain so throughout the study. Zone 3 in green had contamination even though it was well outside our working zone. And the CT value appears to increase over time, or in other words, less RNA was detected from the PCR assay. Feed Samples are, in this graph, feed samples are represented as bars in red from the mixer and purple from the bucket elevator. The lines are represented as swabbed surfaces, orange from the mixer and dark red from the bucket elevator. Note that in the feed, as I reported in the previous podcast presentation, the CT value of the feed rose after the first negative batch was sequenced. Furthermore, with the similar CT values were found in feed as were in the equipment swabs. After the second sequence, there was no detectable RNA in the mixer feed, but there were still low levels detected in the bucket elevator. After sequence 3 and 4, there was no detectable RNA in the mixer or bucket elevator. However, the interesting data from this graph is from the swabs. As we expected, after making the positive batch of feed, the CT level in the swabs and the feed were similar. The surfaces that we contaminated were essentially the same as the feed after the positive batch. However, after sequence 1, the feed PED levels were diluted and the surfaces were contaminated at a similar level after the positive control, that of which were persisted to the end of the study, even when the feed no longer had detectable RNA. As I presented in the earlier podcast, we did not have detectable RNA in the feed after sequence 3 and 4. However, we do have swab surfaces that had dust that contained positive or that had detectable RNA. As this picture on the left-hand side demonstrates, in our boot, the bucket elevator does not completely clean out at the bottom. Remember, in the feed, the samples from the bucket elevator were positive, even after more negative batches of feed were produced from the mixer. This makes sense because there is a potential area of cross-contamination here in the bucket elevator. Also remember from the previous slide, our surfaces of both the mixer and bucket elevator were contaminated even after making multiple batches of negative feed. On the right-hand side, I'll demonstrate how I swab the surfaces, taking the swab from inside the plastic buckets and also along the adjacent belt. In this graph, our results are from the surface type CT values as measured by PCR. And it appears that one pair of boot bottoms had RNA that remained from a previous replication, which was tracked on the concrete floor. However, just to remind you that our feed was negative and remained negative. Our negative batch of feed was negative and remained so. The most important thing to note from this slide is that once a positive batch was made, 100% of the concrete and rubber surfaces were positive, and most of the metal surfaces remained so at the end of the study. Since observing this, we have a subsequent pork board grant that will further evaluate if concrete and metal have an impact on RNA detectability. In our next steps, we are funded by the pork board to conduct bioassay of the swabs later this year. Based on the results from the garage door zone 3, we believe the dust is infective. Thus, practical implications are to minimize and contain dust in the feed mill and dispose of dust instead of adding it to product flow. Finally, I would like to acknowledge our contributors to PED research: the K-State Grain Science, K-State Applied Swine Nutrition Team, the K-State and Iowa State Veterinary Diagnostic Labs, and also appreciation is expressed to the National Pork Board for financial support. And once again, thank you for listening to this podcast.